Okay, so welcome back to this next video in which we are discussing receptor tyrosine kinases. Okay, right, so we now want to turn our attention to the desensitization of receptor tyrosine kinases, which is the mechanism by which the ligand binding to receptor tyrosine kinases and activating them is going to lead to a decrease in the sensitivity of the cell to that ligand. Okay, and the way that this occurs for receptor tyrosine kinases is that they are generally removed from the membrane. Okay, and that means that the cell effectively takes down its way of listening to the ligand. Okay, so if you take off all your receptors for a ligand, then even if the ligand's present in the extracellular fluid, it's not going to have any effect on you. Okay, so you can reduce the effect of a ligand on a cell by reducing the level of receptor tyrosine kinases at the cell membrane. Okay, and this is what's going to happen. So when ligand binds to the ligand binding domains of these receptor tyrosine kinases and causes them to dimerize, we know, of course, now that this causes these phosphotyrosine residues to be produced on the intracellular aspects of the receptor tyrosine kinase dimer. And now we want to study the pathway by which we downregulate the receptor tyrosine kinases. So how do these phosphotyrosine residues on the intracellular aspect cause the removal of this receptor tyrosine kinase from the cell membrane? Well, what's going to happen is a protein called C-cBL, okay? So C-cBL is going to come and bind to phosphotyrosine residues, okay? Like so, on the intracellular aspect of our receptor tyrosine kinase dimer. Okay, now what does C-cBL do? Well, basically, it attaches ubiquitin groups onto the receptor tyrosine kinase. Okay, so where shall I draw this? I'm just going to extend the cytoplasmic tail of my receptor tyrosine kinase so that I have some room to work with. Okay, so what is C-cBL going to do? It's going to attach on a special group onto the side of the receptor tyrosine kinase that is called a ubiquitin group. Okay? So this is a ubiquitin group. Now a single ubiquitin group is going to be added on. Okay, so we call this monoubiquitination. Okay, right. So here in green, this is our single ubiquitin group that has been added on to our receptor tyrosine kinase. Now, what's now going to happen is another protein is going to come and bind to this ubiquitin group that we've got now attached onto our receptor tyrosine kinase here. Okay, so here it comes, binds on here, and to do this it has to have a special domain which is known as a ubiquitin interaction motif. Okay, so this portion in purple that's binding to the ubiquitin group, this is known as a UIM for short, which stands for a ubiquitin, that's the U. Okay, the I is for interaction. Okay, and then the M is for motif. Okay, so that's that pink structure part, well, the pink part of the protein. Okay, so this protein with a ubiquitin interaction motif is going to come and bind to that ubiquitin group that's been attached to the receptor tyrosine kinase. Okay, right. Now, what's an example of a protein with a ubiquitin interaction motif? One example is a protein called epsin. Okay, so this protein epsin, which has a ubiquitin interaction motif, is going to come and bind to this ubiquitin group that's been attached onto the receptor tyrosine kinase by the C-cBL protein. Okay, right. Now, the epsin protein is going to bind to something known as the adapter protein complex 2. Okay, so we'll go aside and just discuss the structure of the adapter protein complex 2. Okay, so this is not a single protein, it's a complex of proteins. And it's usually abbreviated to AP2 for short. So A for adapter, P for protein complex, and then 2. Okay, now the adapter protein complex 2 consists of 4 separate proteins, which I'm going to talk you through now. Okay, so it consists of a main core or trunk domain. Okay, and the main core trunk domain consists of the core portions of the alpha subunit and also of the beta 2 subunits. I'll show these now. Okay, so this portion here, this is the core domain of the alpha subunit. So this is the core portion of the alpha subunit. So I'll colour this in green here. Okay, and the portion that's opposite it, that's supposed to represent the core portion of the beta subunit, or rather the beta 2 subunit. Okay, so this is the core domain of the beta 2 subunit here. Okay, and then in these gaps that I've left in between the core domains of the alpha subunit and the core domain of the beta 2 subunit, we are going to have smaller proteins put inside of those, okay, which are also going to contribute to this full core domain of the adaptor protein complex 2. So firstly, let me colour in the core domain of our beta 2 protein. And by the way, the alpha and beta 2 proteins are going to have other domains which I haven't drawn on yet, so we'll come to those. Okay, and now we've got these two other proteins in here, one which I'll colour in blue and the other which I'll colour in purple here. Now, one of those is what's known as the mu2 protein, so we'll have this one in blue being the mu2 protein, and the other is known as the sigma2 protein. Okay, so this entire structure that we've got here consisting of the core domain of the alpha subunit, the core domain of the beta 2 subunit, and then the mu2 and the sigma2, this is known as the core or the trunk of the adaptor protein complex 2. So this is the core of the adaptor protein complex 2, AP2. Now, each of the alpha and the beta 2 subunits both have these additional domains which stick off them, like so, known as the appendages. So this here, this is the alpha appendage. Okay, and then you also have the beta 2 appendage as well on the other side. And the region in between the core domain and the appendage of an alpha or beta 2 subunit is known as the hinge region. Okay, so here is the alpha 2 appendage, and here is the beta 2 appendage up here, with the hinge region joining the appendages and the core domains. Okay, so now what's going to happen is epsin is going to bind to the appendage of the alpha subunit of the adaptor protein complex 2. Okay, so I'll draw this happening here. So here is our alpha subunit of the adaptor protein complex 2. Here is our beta 2 subunit of the adaptor protein complex 2, again with its appendage coming off up here. And then we have the mu2 and the sigma2 proteins in the middle there. So here is our mu2 protein, and here is our sigma2 protein in pink here. Okay, and then here is our alpha 2 protein in green here, like so. Here's the core or trunk domain of the alpha protein, like so. And then we also have the beta 2 protein, which I'll colour once again in an orange. Here's the beta 2 appendage, the beta 2 hinge, and now here's the beta 2 core or trunk domain in orange here. Okay, right, so that's the adaptor protein complex 2 here. Now, overall then, we've assembled this large complex on the bottom of our receptor tyrosine kinase. This is going to result in the recruitment of Clathrin to the receptor tyrosine kinase. So adaptor protein complex 2s, these will recruit Clathrin. They will bind to Clathrin molecules. Okay, so we are going to get Clathrin recruited to the underside of this receptor tyrosine kinase, and that is going to result in the polymerization of the Clathrin and the formation of an endocytic vesicle that's going to pinch off the membrane that contains this receptor tyrosine kinase and take the receptor tyrosine kinase into an endocytic vesicle. Okay, and to understand this further, we now need to discuss Clathrin in more detail. So I'm going to turn my attention onto Clathrin. Okay, right. So we'll start off with a Clathrin monomer then. Okay, and a Clathrin monomer is going to consist of a Clathrin heavy chain along with a Clathrin light chain. So I'm going to start off by showing you the structure of a Clathrin heavy chain. So this is my picture for a Clathrin heavy chain. It's a single polypeptide. Here's the amino terminus, here's the carboxylic acid terminus. Okay, now this portion that I'm colouring in red here, this is the N-terminal domain of the Clathrin heavy chain. So this is the N-terminal domain. Okay, then after the N-terminal domain, this stretch here, which I'll colour in green, this is going to be the distal domain of a Clathrin heavy chain. Okay, so in green here, this is the distal domain. Okay, then going further towards the carboxylic acid tail, you then have the proximal domain, which we'll colour in orange here. Okay, so this is the proximal domain of our Clathrin heavy chain. And I want to stress we have not shown the full Clathrin monomer yet. Okay, Clathrin monomers are going to consist of not only a Clathrin heavy chain, but also a Clathrin light chain. So what we've got so far is just the Clathrin heavy chain. Okay, we'll come to the Clathrin light chain in a moment. When we dimerize the two together, we'll then have a Clathrin monomer. Okay, right. And this final portion of the Clathrin heavy chain here, shown here in turquoise, this is the trimerization domain of our Clathrin heavy chain. Okay, so we have these four important domains of our Clathrin heavy chain. Now, we're then going to get the proximal domain of our Clathrin heavy chain associating with our Clathrin light chain here. Okay, so this is our Clathrin light chain, which is so called because it's smaller than the Clathrin heavy chain. Okay, and together, if you have a Clathrin heavy chain with a Clathrin light chain, as so, that's then called a Clathrin monomer. So the entire thing is now a Clathrin monomer. Okay, right. Now, Clathrin does not actually exist mainly in the cell in this monomeric form. Instead, it assembles into a trimeric form known as a Clathrin triskelion. Okay, so I want to show you now the structure of a Clathrin triskelion. Okay, so we're going to trimerize 3 Clathrin monomers together to get a Clathrin triskelion. So let's do this. So we'll start by just drawing one Clathrin monomer. Okay, so here's a single Clathrin monomer. Then we're going to attach on via the trimerization domains. Okay, so here's the trimerization domain of our first Clathrin monomer. Here's the trimerization domain of our next Clathrin monomer. Okay, then we're going into the distal domain, then the N-terminal domain, and we need a light chain here. Okay, so let's colour this in to keep us orientated. So here's one Clathrin monomer. Here's the proximal domain of this other Clathrin monomer. Okay, then in green here, we have our distal domain here as well. Okay, our N-terminal domain over here. And then just to add on the light chain now, this portion here and this portion here. So that's only 2 of them now. Let's put on a third one. So here comes our third one. Here is the trimerization domain, then the distal domain, and then the N-terminal domain. Okay, so once again, the trimerization domain there in turquoise, the proximal domain there in orange, okay, the distal domain here in green, and then in red, here is our N-terminal domain. And then associated with the proximal domain, you then have a light chain. Okay, so this is the structure of a Clathrin triskelion then. You trimerize 3 Clathrin monomers together to get a Clathrin triskelion, and this is the form that Clathrin generally exists in within the cell. Okay, so a molecule of Clathrin will come and associate with our, wherever we've gone, with our adapter protein complex 2 here. Okay, so I'll just show this like so. So here, well, it actually associates with the hinge domain up here of the beta-2 appendage, so I shouldn't show it there. Okay, so how can I show this? I'll try and show my Clathrin triskelion here. Okay, so here is my Clathrin triskelion. Like so. Right, there we go. It's going to come and associate with the appendage, sorry, the hinge of the beta-2 subunit here. Okay, and then what's going to happen is it's going to start polymerizing. Now when Clathrin triskelions polymerize, they form a structure that is very similar to a buckyball. Okay, so I want to remind you of what a buckyball looks like. Now, if you are familiar with the structure of football, then you already know everything you need to know, basically. Or if you're an American, a soccer ball rather than a football ball. Basically, footballs or soccer balls, their structure is the exact buckyball that Clathrin triskelions are going to assemble into, basically. It's also the structure of the chemical Buckminster fullerene, okay, which is that 60-carbon ball, basically. So let me just remind you of this. I'm going to attempt to draw this. Okay, I'm only going to draw part of it. I can't draw the full thing. Let's imagine that, well, this buckyball is going to be made out of a tessellation of regular pentagons and also hexagons. So let's imagine that we'll start with a regular pentagon here. Okay, like so. So here's a regular pentagon. And let's imagine this one is in the plane of the piece of paper. Now let's imagine we want a ball, we want to build a buckyball. Okay, so what we're going to do is we're going to attach regular hexagons onto this side, this side, this side, this side, and this side. Now we can't keep them in the plane of the piece of paper, because if we do that, okay, what we'll get is something like this. If we were to put the regular hexagons in the plane of the piece of paper, we'd get something that looked like this. Okay, the regular hexagons would not join together perfectly. There would be this gap in between them. Okay, and this would happen at every single one. Okay, so to get the regular hexagons to join together, what you have to do is you have to lift all of them out of the plane of the piece of paper. Okay, so here are our regular hexagons that I've all attached. What you're going to do is bring this one up, tilt this one up, tilt this one up, tilt this one up, tilt this one up, and they're going to start forming a ball, basically, or a bowl, rather, is what I meant. Okay, so continue having the pentagon, then, in the plane of the piece of paper here. Okay, and now we're tilting these regular hexagons out of the plane of the piece of paper towards us. Okay, like so. And they're starting to form this bowl-like structure. Sorry, I didn't mean ball. It's not a ball yet. Okay. So these hexagons are all tilted out of the plane of the piece of paper towards us. And now you're going to continue on building this bowl. You're going to insert in regular pentagons here. And these will almost be vertical now. They'll be tilted very much so out of the plane of the piece of paper. Okay, you'll put in a regular hexagon here, a regular hexagon here, a regular hexagon here, and then also a regular hexagon here. Okay, and then around each of these regular hexagons, you're now going to have around each of these regular pentagons, you're now going to have 5 regular hexagons around those, okay, and it will continue going on, basically, and this will fold around into a 3-dimensional ball. Okay, so that's what these Clathrin triskelions are going to produce, basically. Okay, now it's not going to quite work beautifully. You might think, well, is it going to work ridiculously beautifully that we're going to put Clathrin molecules on here and they're perfectly going to tessellate? And the answer is no. You have to make the Clathrin triskelions overlap. So let me show you how we're going to do this. So I'm going to draw a Clathrin triskelion here. Okay, so here's one of these Clathrin heavy chains here. Here's another one of these Clathrin heavy chains, and here's another one. Okay, and I'll mark on the light chains as well. So here's a light chain, here's a light chain, and here's a light chain. Now, you can put other Clathrin triskelions onto this as well. So we're going to put loads of these Clathrin triskelions together and make one of these buckyballs. Now, let's say we'll put another one here. Okay, so I'll colour this one in red. So this one isn't going to not overlap with the first, basically. This Clathrin heavy chain here is going to overlap heavily with this one. This one will then go up there, this one will go up here. Okay, so basically the Clathrin triskelions don't not overlap. Okay, they do overlap. So it's not a perfect tessellation, but the idea is that you put lots of these Clathrin triskelions together, and they overall build you a shape that is one of these buckyballs. Okay, so that's what's going to happen. we are going to be assembling one of these Clathrin triskelion buckyballs underneath our receptor tyrosine kinase here. So one of these is going to start forming underneath here. Now, what's the effect of that? Well, if you imagine, what we have effectively done is if we just draw our receptor tyrosine kinase dimer as a blob for now, to keep it nice and simple, we have put loads of proteins in between them, but effectively what we have done is indirectly attached our Clathrin onto the bottom of the receptor tyrosine kinase. Yes, it's indirect, but that's how we're going to simplify it for this picture. So this, in purple, this represents our receptor tyrosine kinase dimer. Okay, and in blue here, this represents our Clathrin. Okay, so they're not directly attached, but there's lots of proteins in between them. But to keep it simple, they are indirectly attached, so we'll show them attached. Okay, what's now going to happen is these Clathrin molecules are going to start assembling into one of these bucky balls, and the effect of that is that the membrane is going to bend around the assembling bucky ball. Okay, like so. So if this is the phospholipid bilayer here, represented by these two lines, we are going to invaginate the membrane as we build our buckyball from these Clathrin molecules. Okay, so here is the Clathrin buckyball assembling here. So this is our polymerization of Clathrin here. Okay, and it's pulling the receptor tyrosine kinase down, or rather it's pushing the rest of the membrane upwards. Okay, so these are loads and loads of Clathrin triskelions, which are all polymerizing together to make this buckyball shape, which is invaginating the membrane downwards here. So in purple, this is our receptor tyrosine kinase dimer here. Okay, and let me just reassemble this. Okay, then we've got our Clathrin triskelion buckyball being assembled here. Okay, right, and what's going to happen is this Clathrin triskelion buckyball is going to complete, basically. Okay, so it's going to complete its way round, and then it will pinch off the membrane as it does so. Okay, so you'll close off this gap that you've got here that currently connects this space here with the extracellular fluid, and what you'll get is this portion of the membrane here will reseal with this portion. Okay, so this will reseal with this portion, and then you'll have this vesicle that's been pinched off from the membrane. Now I'm going to demote the plasma membrane from being represented by two lines to being represented by just one line to make the picture simpler. Okay, so the membrane's going to reclose up. So this is the two green portions having reclosed up. And you're then going to have this vesicle pinched off from the plasma membrane like so, which will have our receptor tyrosine kinase dimer within it. Okay, here in purple, and then at the moment it will be surrounded by this Clathrin buckyball here. Okay, now whilst you are surrounded by a Clathrin buckyball, you can't actually do anything. Okay, you can't fuse with any membranes of the intracellular organelles. Okay, so the first stage is going to be to remove this Clathrin coat from this endocytic vesicle. Okay, so this membrane-bound little structure that we have pinched off from the cell membrane, this is called an endocytic vesicle. Okay, and the first thing that's going to happen to it once it's been pinched off like so is that we're going to uncoat the Clathrin. Okay, so the first thing that happens is uncoating then. Okay, in which the Clathrin coat is going to come off, and now what we've got is our endocytic vesicle here with our receptor tyrosine kinase dimer here, which I'll colour in purple. Okay, right. So now, this endocytic vesicle that's no longer coated in Clathrin like so, this is going to go off to intracellular organelles. Okay, so the first intracellular organelle it goes to is something called the early endosome. Now, the early endosome is not an endocytic vesicle. People often confuse endocytic vesicles and endosomes. Endosomes are intracellular organelles, okay, and there are two main types of endosome. There is an early endosome and there is a late endosome. The first one you go to is the early endosome. To put this in perspective, if we draw a cell here, the early endosome is a large intracellular organelle here, whereas an endocytic vesicle is a tiny little speck on this picture, basically. This thing is tiny compared to this. This is a huge, great, membrane-bound intracellular organelle, okay? So some people will tell you that these things that you are pinching off from the membrane, that those are endosomes. They are not endosomes, they are endocytic vesicles. Okay, the endosomes are intracellular organelles. Okay, the problem is that people try to oversimplify these pathways and they show the receptor tyrosine kinase instantly going into an endosome, and they miss this shuttling between the plasma membrane and the early endosome, which occurs through these tiny little structures known as endocytic vesicles. Okay, so what's going to happen then now is our endocytic vesicle is going to go and fuse with the membrane of the early endosome, and then our receptor tyrosine kinase dimer will end up in the early endosome. Okay, now potentially it can just sit in the early endosome, okay, and also, sometimes they can be recycled back to the cell membrane, so they can pinch off again into a vesicle which will take it back to the cell membrane. However, often what's going to happen is you're then going to pinch off a vesicle from the early endosome, okay, which will contain our receptor tyrosine kinase dimer here again, okay, and this is now going to go off to another endosome. Okay, and the endosome it's going to go to is the late endosome. Okay, so this is another large intracellular organelle, another one of these endosomes. Okay, again, what will happen is the endocytic, well, it's no longer an endocytic vesicle, it's a vesicle now. This vesicle that's trafficking the receptor tyrosine kinase from the early endosome to the late endosome will fuse with the late endosome membrane, and that will put the receptor tyrosine kinase into the late endosome. Okay, finally, if we want to destroy the receptor tyrosine kinase, you'll get a vesicle budding off the late endosome, shown here, which will then go to another intracellular organelle known as a lysosome, okay, which I'll show here, and this vesicle will fuse with the lysosome, and the receptor tyrosine kinase will be broken down in the lysosome, so there are many enzymes which will break the receptor tyrosine kinase down within the lysosome. Okay, so that's the pathway by which we can remove receptor tyrosine kinase from the membrane and thereby reduce the sensitivity of our cell to the ligand for that receptor tyrosine kinase. So that now concludes our discussion of receptor tyrosine kinases.